This is Lecture 4, How Genes Work, Part 2. So what we're going to talk about further is this idea of the roles of transcription and translation, and we're going to introduce the idea of the genetic code. Alright, so here we see the central dogma. It's just illustrated a little bit differently than in the previous Part 1 slide. So we're seeing the flow of genetic information from DNA here to proteins here. Now there are two terms that are really important here, and the two terms are genotype and phenotype. Genotype is the genetic information held by an organism. Phenotype is the measurable physical properties that are the result of genotype. So when you look at that forest mouse in the bottom photo, you see a small brownish mouse, and he has a particular shape of eyes, a particular size, a particular shape of ears. We could weigh the mouse, we could measure the mouse, we could describe the color, so we could measure him in terms of qualitative terms. We could even quantify a lot of those things. We could look at the melanin or pigment in his fur, and we could measure it and give a number to it, but those are his the qualities that determine his phenotype. That is the result of him using his genotype, all of his genetic information, in order to make proteins, and those proteins then do the work of cells and the work of the organism, resulting in phenotype. So here we see a gene up here, stored in DNA, the informational molecule. It's transcribed through the act of transcription to a molecule of messenger RNA, and then that messenger RNA is translated, and that is done by the ribosomes, and that translation results in a polypeptide, protein primary sequence. That protein primary sequence then goes ahead and assumes secondary and tertiary, and in some cases quaternary structure. You get a functional protein, the proteins do their work, and the result of that work are measurable physical properties known as phenotype. So when you talk about the phenotype of an organism, you might refer to something very, very specific. For example, does the mouse have brown eyes, or does the mouse have, I don't know, blue eyes? Or you might just talk about something like enzymatic activity. You might talk about the level of activity of the mouse's lactase digestive enzyme, one versus another. Or you might talk about phenotype in more general terms, but there'll be something discernible, something physical. All of that information is ultimately encoded in the mouse's genotype. While we're looking at this slide, let's remind ourselves that the act of transcription is done by RNA polymerase. It's an enzyme, and the act of translation is done by structures known as ribosomes. In a eukaryotic cell, transcription happens in the nucleus, and translation happens outside of the nucleus at the ribosomes. In a prokaryotic cell, they both happen at the chromosome. Alright, so if you look at two different mice, and this is a beach mouse, but let's just imagine they're the same species, and this mouse looks quite different. So this mouse has a different phenotype than the previous mouse. So that means that it must have one or more genes that differ than the previous mouse. They don't differ in their identity, they differ in their sequences. So that means that they have different alleles. An allele is a form of a gene, and we defined gene in the previous part of this lecture. Differences in genotype result in differences in phenotype. So very slight changes in the genetic code of an organism can alter their physical traits and their discernible phenotypes. I also want to point out that not all genes encode proteins. Okay? A lot of genes encode functional RNA. So functional RNA could be transfer RNA, as shown here, or it could be rRNA, which makes up part of the ribosome. So those structures have genes that encode them. Those genes are transcribed, but then they are not translated. Instead, we have the result of RNA. That RNA assumes some secondary structure. Examples of the secondary structure it might assume are these internal base pairings and these hairpin loops shown here in this tRNA molecule, and the result is what is known as a functional RNA. In that case, transcription is important, but not translation. All right, so let's talk about the genetic code. There's some terminology here that matters, and one of the terms is codon. So a codon is 3 nucleotides that represent a specific amino acid. Codons can be written in DNA nucleotides or RNA nucleotides. Okay, so when we're thinking about the genetic code, it's also important to understand what are known as the DNA-RNA base pairing rules. So those rules relate DNA sequence to RNA codons, and the DNA-RNA base pairing rules are this. Okay. The 3 possible nucleotides of DNA are A, T, C, and G. Those correspond to RNA nucleotides. A corresponds to U, T corresponds to A, C corresponds to G, and G corresponds to C. So when you take DNA and you transcribe it, the act of transcription, which is done by RNA polymerase, then wherever in a gene of DNA there is an A, you place a U in the RNA. Wherever there's a T in the DNA, you place an A in the RNA. A C, you place a G you place a C, and so on. You need to understand those base pairing rules in order to understand transcription. You need to understand the role of transcription and how it works in order to understand the flow of genetic information, how the genetic code works. So the codons that are found that are important in cells and in the genetic flow of the genetic message are typically expressed in what is known as a codon table. Standard codon tables are written in the language of RNA. That's why you see U's there. So you'll notice, let's look up here on the top left. Phenylalanine is an amino acid. So all of that typing in black signifies the 20 common amino acids. There are 2 codons that represent phenylalanine, UUU and UUC. Okay, so the UUU is the RNA codon for phenylalanine. Okay, it relates to, or it is derived from, a DNA codon that was A. All right, you'll notice that many of the amino acids have more than one possible codon. So this is what we mean when we talk about the genetic code being redundant. So UUU means phenylalanine, so does UUC. Similarly, leucine, shown below the phenylalanine. UUA means phenylalanine, UUG means leucine, and UUG means leucine. Most of the amino acids have more than one possible codon. One thing you'll notice is that they only differ at the 3rd position. So if you look down here at valine, all of the codons for valine start with a G and a U, and they only vary at the 3rd position. and that's very typical, and it relates to a concept known as wobble, which I won't really get into right now, but we'll discuss later in the semester. I also want to point out, however, that the genetic code is very specific. So UUU always means phenylalanine, never any other amino acid. While we're looking at this table, let's notice a couple of other features. Methionine is represented by the codon AUG. Methionine is also known as the start codon. So all polypeptides begin with the amino acid methionine. And when we get into talking about translation, we'll talk a little bit more about the details of that process. But AUG is known as the start codon, and it also always signifies the amino acid methionine. Also note up here, UGA, UAA, and UAG all signify stop. So when a ribosome reaches those codons on messenger RNA, it knows that the polypeptide is done and it does not incorporate any more amino acids.